Hi, everybody. Thank you very much for dialing into the investor presentation today. I'm delighted that we'll be speaking with the inventor or developer of our iNKT cell platform, Professor Karadimitris. And we're excited to be presenting today because we're going to talk about some of the key data that was presented at the 65th Annual ASH Conference in San Diego, California. And let me just jump straight in. So just a quick refresher on our CAR-iNKT cell manufacturing process. It is our platform that we utilize to make our cell therapies that we're using to target initially blood cancers, but also looking towards solid tumors. So that was licensed from Imperial College London after being developed for some time by Professor Karadimitris from Imperial College London in 2021. So importantly, that platform is patent protected with long patent life out until 2038. And it is granted already in Europe with applications also pending in the US, Canada, China, Hong Kong, and also Australia. So a key benefit for our platform is we do see high transduction efficiency of the chimeric antigen receptor, or CAR, and that helps to ensure that many of the cells actually contain the ability to kill the cancer cells. So by doing that, we increase potency. And because we actually perform that particular step on lower iNKT cell numbers, it actually minimizes the costs required for quite an expensive product that we use to make those cells. And importantly, over the journey, what we've been able to show is when we use our manufacturing process and start to scale up using clinical grade reagents, that we actually see that we retain the associated tumor killing, which is what we would like to see. And we are able to produce multiple doses per batch, which is very important for an off-the-shelf therapeutic. Now, in terms of the production itself, very quickly, the way it works in contrast to a CAR-T, where it starts from the patient and finishes with the patient, we collect blood from a healthy donor, we isolate their iNKT cells, and then we engineer iNKT cells to produce a CAR, and that will be something that we, as I referenced earlier, is like a guided missile that will draw these cells towards the cancer to eliminate their destruction. They get grown up in a lab into the billions before being vialed, frozen, and then when a patient is eligible to take that therapy, they will be thought out and delivered to those patients. So some key advantages are healthier starting material because the starting material is a healthy donor, so they haven't seen a cancer or chemotherapeutic interventions and so forth. We do think it's very important that we'll have multiple doses per batch, which of course then provides scale. Now, for patients that have particularly aggressive disease, faster access to treatment is paramount. And so we see that that would be the case by pulling this out of the freezer as opposed to being manufactured in real time. And because it's sitting in the freezer, it essentially completely removes the risk of manufacturing run failure at the point that it has been frozen. Now, I said I'm delighted that we've got Professor Karadimitris on the call today because he has spent a significant proportion of his career understanding iNKT cell biology and, of course, what makes them so useful for a candidate in a cell therapy to treat cancer. And just a quick refresher, there are many immune cells in our body, and the ones in the middle, they're the iNKT cells, is what we're most interested in. For a number of reasons, we see them as so important because they are the front line of the human immune system, bridging both the innate or the fast acting and the adaptive, which is the longer term or more durable immune responses. They actually can kill like T cells and NK cells, and they naturally target and kill certain cancers that actually have CD1D already on their surface. And really importantly, for an off-the-shelf therapy, we know that they don't cause graft versus host disease. And with some of Professor Karadimitris' pioneering work that demonstrated not only that they don't cause graft versus host disease, but they actually can prevent it in some settings. And there are, of course, a number of other properties, the fact that they also influence pro-cancer cells like tumor-associated macrophages or myeloid-derived suppressor cells. And they can also infiltrate tumors and secrete or release other components that start to engage other immune cells to get involved in the fight against the cancer cells. So just very briefly on the blood cancer market and the relationship to cell therapies, we have seen a pretty large explosion of the field over the last, say, 5 years or so. There are now 4 approved CAR-T products targeting CD19. In 2023, they'll be expected to have more than $2 billion in revenue. They are already moving into second-line therapy for some of them, which is great to see. But as of today, there are still no approved off-the-shelf CD19 targeting cell therapies. And for multiple myeloma, another form of blood cancer, we've seen slightly slower development with the first approved product, and there are now 2 available. The first approved product was in the US in 2021. Are expecting significant revenues for those 2 products across 2023, but similar to the CD19 targeting products, there are still no off-the-shelf cell therapies to target multiple myeloma. So with that, I'd like to just hand it over to Professor Karadimitris, and he will start to talk through some of the key data that was presented at a poster this week and at the ASH conference in San Diego, California. And thanks very much, Tassos. Go for it.
Thank you, Michael, and thank you for inviting me to present the key findings of work we've been conducting for the last couple of years in the lab, trying to understand what co-stimulatory domain will be best suited for treatment, CAR-iNKT treatment of multiple myeloma. Of course, the co-stimulatory domains can be modified and exchanged, and the purpose is to drive increased activity of the CARs and CAR-Ts or CAR-iNKTs in our case. So we set out to compare side by side 5 BCMA targeting CARs with different co-stimulatory domains, the most commonly used co-stimulatory domains. So we generated the appropriate constructs and we tested them side by side, as I said, both in vivo as well as in vitro. The in vitro studies, which we're not showing here, demonstrated clearly that one of the 5, that is the second generation CD28 co-stimulatory domain, was superior compared to the others in terms of avidity. And avidity basically measures how tight a cell like CAR-iNKT cells bind to their targets, in our case, myeloma cells. And recent data suggested that avidity can predict the in vivo anti-tumor activity of CAR-T, or in our case, CAR-iNKT cells. So therefore, what we did was to establish a multiple myeloma model in mice, in immunocompromised mice. And in these mice, we transferred CAR-iNKT cells that have 5 different endodomains, co-stimulatory domains. Those that we call second generation had a single one and were made of either of CD28, OX40, or 4-1BB, and 2 third generation that combined 2 of these co-stimulatory domains. One was the CD28 4-1BB combination, the other was the CD28 OX40 combination. So we treated these mice deliberately with a relatively small dose of anti-BCMA CAR-iNKT cells, because if we gave them a high dose, say of 10 million, then all of them, because of the big numbers, would be expected to perform reasonably well. But by stressing the system using a small number of CAR-iNKT cells, we could tease out the ones that had really the inherent ability to perform better. And after more than 41 days of following up these mice, only the mice that were treated with a CD28 containing co-stimulatory domain with CAR-iNKT cells containing the CD28 co-stimulatory domain remained alive. And we're not showing here the majority of these mice, and there were about 7 and 8 in each group, had very little, if any, evidence of myeloma in the bone marrow, in the spleen. Therefore, as predicted by our in vitro avidity studies, in vivo studies indeed confirm that CD28 co-stimulatory domain outperform all the other combinations.
And Tassos, I might have a few questions here. So one is, when we look at the current products or cell therapies that have been used to treat multiple myeloma, none of them yet contain a CD28 co-stimulatory domain. Is that correct?
This is correct. And as a matter of fact, I'm somewhat surprised that we don't know why both of them has a 4-1BB. I have not seen any data from the manufacturers of these very successful products, but still falling short of achieving long-term remissions and cures, what was the basis or the rationale of using 4-1BB in the commercial product? I can only speculate that they were guided by the credible data, in vitro data and in vivo data in animals, preclinical models, and also in humans, that indeed 4-1BB CAR T cells appear to persist longer than CD28 CAR T cells. And that, as I said, has been seen in clinical practice.
Thanks, Tassos. And another question relating to multiple myeloma itself. That's something that's been quite an area of interest for you, both professionally and personally. And can you perhaps comment on why you think iNKT cells and this data with the BCMA CAR are so important for a condition like multiple myeloma?
Yes. First, let me say that I'm the director of the Langmuir Center for Multiple Myeloma Research, and we're very proud that we focus in a concerted manner in investigating the biology and therapeutic applications for this still incurable blood cancer. And of course, myeloma is also almost a prototype of a disease, of a cancer, which lends itself to the study of the therapeutic applications of iNKT cells, because myeloma plasma cells, as their normal counterparts, express CD1D. And in fact, we showed that many years ago, and we also published work that shows that CD1D is, in a way, a pro-survival factor for multiple myeloma. So the fact that iNKT cells, of course, they can engage with tumor cells like myeloma through the invariant TCR and its interaction with the CD1D, and that adds to the anti-tumor effect of iNKT cells, be it as CAR-iNKT or TCR-iNKT, makes really our approach even more important, and raises our hopes that will provide something that is missing from the current cellular immunotherapies of multiple myeloma.
Yeah, agreed. And I think then in the context of having a product that we would like to utilize off the shelf, having the ability to have something that's dual targeting, going after CD1D and potentially BCMA as well with the CAR is of paramount importance, I think, for those off-the-shelf type products. So Tassos, can you talk more about why you think the CD28 co-stimulatory domain performs the way it does, especially relative to the others, and what this could mean in terms of a therapeutic strategy for an iNKT cell platform or program, particularly in the context of something being off the shelf?
Well, we are investigating the underlying mechanism, and our data so far, which actually have been based on a comparative analysis of the transcriptome of the different products that we made with a different costimulatory domain, shows that it has likely to do, but we're still working on it, with the induction by the CD28 CAR of a number of surface molecules that, if proven, if their case is proven in experimental work, would increase the interaction, the avidity of CD28 iNKT with myeloma cells. So there's something inherent in myeloma plasma cells and something inherent in the ability of CD28 to induce ligands for these myeloma-specific molecules that makes CD28 the most effective costimulatory domain, specifically for multiple myeloma. Of course, we know it is more effective in the context of lymphoma too, but that highlights the point that in order to develop the most effective CAR constructs and eventually CAR-T or CAR-iNKT or CAR-NK cells, you need to study in a comparative fashion the different costimulatory domains and their combinations in the context of the particular cancer that you're interested in.
Great, thanks, Tassos. So that's really interesting, I think, and it's a great point that making clear that when we do look to create a new therapeutic, that we are investigating head to head the different domains that can be switched out. So we actually are selecting what we would consider to be the best performing product moving forward. That's very interesting. Well, excellent to hear from you again, Tassos. We really appreciate your time, and we're really looking forward, obviously, to taking Arovella's lead product, ALA-101, into clinic alongside you and your team. But just a quick refresher for everybody. As I said, we do have a novel iNKT cell platform that consists of a unique and what we would consider very robust manufacturing pathway. And what we've seen today is that the data demonstrates that clearly BCMA CAR-iNKT cells can effectively kill myeloma tube cells and prolong animal survival. And so this supports the notion that even though we're developing our first program for CD19 positive blood cancers, there is the opportunity to expand this for use in multiple tumor types. I think we also saw quite clearly that when selecting a particular cell type and a CAR against a particular cancer, how important it is to assess the different co-stimulatory domains. And it was pretty clear here that the CD28 outperformed other co-stimulatory domains that have been used in the past. And it is worth noting here also that for our lead program, ALA-101, we have selected the CD28 co-stimulatory domain as well after some internal analysis on that. And just to finish, I'd say it's also interesting that both of the BCMA targeting CAR-T products do contain the 4-1BB co-stimulatory domain. So we'd be very interested to see how an iNKT cell product in this space would perform. And just to finalize here, I'd just like to say that we do believe that our iNKT cell therapy platform it's a very exciting, novel, off-the-shelf therapeutic strategy. It's unique globally and locally, and we do hope that it will provide improved patient access. And finally, Tassos, we're very excited that I think there's more data to come to be presented from your lab at ASH and then in the coming days. So good luck for that, and looking forward to continuing to provide updates as we move forward. And thank you again very much for your time. I appreciate you joining.
Good to see you, Michael, and everyone in Arovella. Thank you.
Thank you.